I'd like to spend some time this morning talking to you a little bit about the work that we've been doing in my lab to understand sex chromosome evolution in the wild strawberry genus. So as you're probably going to hear a lot about today, there's a wide variety of sexual system and sex determination systems across the tree of life, and ranging from small genic determination to environmental determination, or chromosomal determination where either females or males can be the heterogametic sex. And I'm sure lots of people will be telling you about many of these diverse systems today. So along the lines of understanding how sex chromosomes come to be, there's a classic theory that suggests that they've evolved from autosomes by acquiring a sex-determining region that then has suppressed recombination around it that initiates the process of heteromorphism. And much of the early work done on sex chromosomes was done in some very heteromorphic and very old systems like those in humans and in chickens. But more recently, work has focused on systems that have homomorphic sex chromosomes, so ones that cytogenetically look identical, and may have very small non-recombining regions. And the idea was that these potentially were younger systems. But recent work has suggested that this progression may not always evolve in this particular direction, and that sex-determining regions and sex chromosomes potentially can be one of the most dynamic sets of chromosomes within the genome. In fact, really fascinating work in sticklebacks has shown that different chromosomes can become the sex-determining chromosome. Others that have shown that sex-determining systems can move from one chromosome to the next and result in turnovers in the chromosome that determines sex, and that the previous sex chromosomes can even revert back to autosomes. And so many of the people who've done this seminal work are in our program today, and I'll leave it to them to talk more about those systems. But what you might notice here is that I'm not showing any plants in this diagram. And in fact, most of this work has been done in animal systems. But I'd like to talk to you today a little bit about why I think flowering plants can provide some novel insight into this conversation about how sex chromosomes evolve. So in particular, flowering plants, the ancestral state is hermaphroditism, where every individual is both male and female. And the evolution of separate sexes, or is referred to as dioecy, has occurred repeatedly in plant evolutionary history. In fact, while dioecy only represents about 6% of the 300,000 flowering plants, it's really distributed very widely across those families, existing in about half of all the flowering plant families. So this is really interesting because it suggests that there's some strong selective pressures for having the separation of the sexes. In fact, some recent estimates by Suzanne Renner has suggested that there's been at least 800 and possibly as many as 5,000 independent origins of the separation of sexes in flowering plants, providing us a really important framework for studying this kind of comparative work. So amongst those estimated 15,000 dioecious plant species, interestingly, we only have cytogenetic or genetic evidence for sex chromosomes in 40 of those, with about half of them showing the kind of heteromorphism that we come to expect in animal systems. And so here's some of those examples of highly heteromorphic sex chromosomes in the genus Silene, or Rumex, or Coccinea. And we also have homomorphic systems that have much smaller non-recombining regions. But one thing that you'll probably be able to see here is that there's really not a very good correlation between the age of the system and the size of that sex chromosome. So for instance, Coccinea, which actually has some of the most heteromorphic sex chromosomes in flowering plants, is of equivalent age to papaya, which has a very small sex-determining region. And recently in the date palm, evidence that this system might have been around for 50 million years but has a very small sex-determining region. So there is some strong interest in understanding what might be promoting this diversity and lack of correspondence between the age and the stage of the sex chromosome. So one of the ways that we might think about this is if we think more closely at how sex determination occurs in flowering plants. It turns out that there are multiple different evolutionary pathways that can lead from hermaphroditism to dioecy. One we refer to as the Dimorphic Pathway, and this involves the initial invasion of a hermaphroditic population by a unisexual individual, and that individual could be male or female. The monomorphic pathway addresses how there might be variation in the ratio of male to female flowers on a plant with gradual progression to being a unisexual individual. So in addition to these multiple different pathways, there's also multiple genetic avenues for sex chromosome formation amongst them. So even if we simply think about the dimorphic pathway going from hermaphroditic individual to separate sexes, there's multiple different ways that that chromosome could evolve depending on whether the first mutation is a loss of male function or a loss of female function, and the dominance relationships of those. So the end result could either be ZW or XY sex chromosomes. So I want to just focus in for a moment on one of these particular pathways, which is from hermaphroditism to dioecy through gynodioecy, and talk a little bit about the theory that underlies how that pathway might come about. So here I'm illustrating the canonical two-gene model of sex chromosome evolution, first put forth by the Charlesworths, and I'm showing this in cartoon form with the end result being ZW sex chromosomes because that becomes relevant later on in my talk. So the idea being that you have a hermaphrodite individual with an autosome that acquires a male sterility mutation, and here that male sterility mutation is dominant, producing a female individual, and then we will have a population that we refer to as gynodioecious. The second mutation that occurs has to have complementary dominance relationships. So here's a female sterility mutation that's recessive, and it evades that gynodioecious population, reducing fruiting ability of the hermaphrodites, and when in a homozygous state produces a male individual. So when we have both of those mutations segregating in a population, we have the potential for 3 different morphs, which is a breeding system known as subdioecy. There's strong selection for the linkage between these 2 mutations because in addition to the 3 fertile morphs that I show you here, there's a 4th type that can be produced with these 2 mutations, and that's an individual that acquires both the male sterility mutation and is homozygous for the female sterility mutation, and thus is a neuter individual. So there'd be strong selection to limit the production of those neuters, and therefore selection to enhance the linkage and reduce recombination between those 2 loci. As a result of that recombination suppression, there's the potential for expansion of that area where male sterility is linked to female fertility, producing the sex-determining gametolog, the W in the CW system. And through progressive rounds of recombination suppression, we might find either expansion initially and then degeneration of that non-recombining chromosome, and ultimately dioecy in this system. So we're interested in understanding whether or not this kind of patterns could be seen in flowering plants. And we've been working in a genus Fragaria, which is the wild strawberry genus, first to address questions about those early stages from autosomes to sex chromosomes and whether or not we see evidence of that initiation of recombination suppression, and then also expanding out to better understand whether we see this evolving multiple times within the group or if there have been turnovers in the sex-determining chromosome. So a little bit about the strawberry genus. It's relatively young, less than 4 million years old, and it has about 27 different species in it. You might be familiar with some of those species because 2 of them are responsible for producing the cultivated strawberry. And recently, there's been a draft genome produced of one of the diploids in this group. But pretty much once you've seen one strawberry, you've seen them all. They're all very similar in their vegetative form, their flowers, and their fruit types. But the reason why strawberry is a really good system for studying questions about sex chromosome evolution is because within this small genus, there are all the different sexual systems that I just mentioned to you, from hermaphroditism to full dioecy, scattered throughout this tree phylogeny of the genus. And what we know is that there have been at least 4 and possibly 5 different transitions from hermaphroditism to sexual polymorphism within this group, providing us a lot of opportunity for comparative analyses. So the questions that I want to address today briefly is whether or not we have evidence of this 2-linked loci within this group, whether we can see the initiation of recombination suppression when we compare among some species, and then secondarily, whether there's other kinds of dynamics going on in the sex-determining region. Was it initiated once or potentially multiple times? And can we see evidence of whether or not the stage of the sex chromosome is reflected in the age of the species? So the work that we've been doing is focusing on a smaller clade within the genus, which is referred to as the Octopod Clade. It's less than 2 million years old. And why I refer to it as the Octopod Clade is because polyploidization is an important speciation mechanism in this group, and the members of this group are octoploids, although they have disomic inheritance. So for your purposes, you really just need to know they have quite a lot of chromosomes, and when we are looking at one autosome in a diploid, we're actually going to see 4 of those when looking at an octoploid. So when we started this work, the 2 species of interest, Fragaria chiloensis and Fragaria virginiana, were thought to be sister species, but now with greater resolution of a whole plastome tree, we now can see that Fragaria virginiana is ancestral to Fragaria chiloensis. Okay. So those are the 2 species I'm going to be talking about more. Fragaria virginiana is distributed widely throughout North America. It exists in meadows and rural habitats. It has a subdioecious breeding system, so we have females, hermaphrodites, and males in it. In contrast, Fragaria chiloensis is a dioecious species. It is restricted to coastal areas in North America and also South America. It's known as the beach strawberry and has stronger sexual dimorphism. So we have taken a 3-pronged approach for addressing these questions. The first has to do with using genetic mapping and phenotyping to identify where the sex-determining region is within the genome. And our original maps were using microsatellites, but now we've been using targeted sequence capture to get really high-resolution chromosome ideas about what the chromosome composition is. This also allows us to get a sequence that we can use for phylogenetically mapping the chromosomes. As I said, we're dealing with an octoploid, so it's of interest to us to understand the ancestral relationships of the chromosomes or subgenomes that contribute to that octoploid. And we can do that through phylogenetic analysis of those chromosomes. And lastly, we've started to use whole genome sequencing to identify female-specific sequence to characterize the sex-determining region. And so to quickly move on to the questions, first dealing with whether or not we have 2-linked loci or evidence of recombination suppression. We started by mapping the sex-determining region in the species Fragaria virginiana. And here I show you 4 chromosomes in the homologous group 6 from just the maternal map. And what we found is that we had male sterility being dominant, and it was linked to female fertility. And we found recombination between those 2. The reason why we know that there was recombination between them is because this cross produced a characteristic phenotype, individuals that were neuter, i.e. male and female sterile. So that single location explained 94% of the variation in sex within this cross, and we suggested that that would reflect a proto-ZW chromosome. When comparing the sex-determining region in Fragaria chiloensis, the dioecious species then, interestingly, we also found that male sterility was dominant and linked to female fertility. But in this cross, we did not find any of these phenotypic recombinants, and so we suggested that that recombination between these might be lower. Since that time, we have used the targeted sequence capture to get much better genomic resolution and sequence SNPs that we can characterize recombination in this region. And from that data, we now know that there is a very small sex-determining region which has suppressed recombination, where the recombination in the mother was almost half or less than half of that in the father. So we now know that we do have two linked loci in Fragaria virginiana, two linked loci in chiloensis with less recombination between them. But the thing that was most striking possibly between these two species was that although they're both on chromosomes in homologous group 6, they seem to be different chromosomal locations. So we're really interested then in determining whether or not homologous group 6 was a hotbed for sex chromosome evolution, or whether or not it had been evolved once, sex determination had evolved once and been moved among these chromosomes. So in order to address those questions, we needed to know a little bit more about the chromosomes within homologous group 6. And so, so what we did here is taking the 4 chromosomes from the mother and the father from each of those crosses, we created a phylogenetic tree where the tips on this tree are actually those chromosomes, and along with the diploid sequence from some of the other members of that genus. And what you can see here is that those chromosomes form 4 clades, one of them mapping with Fragaria vescobracteata, which is a known maternal contributor to the octoploid, one grouping with Fragaria inumi, which is also a putative paternal contributor to the octoploid, and then 2 that are in this clade, but we don't have a living relative that's very closely related to them. So this helps us characterize the genesis of the chromosomes within that octoploid species. So we can now with confidence say that we have different chromosomes housing the sex-determining region. In virginiana, it's referred to now as B2. In chiloensis, it's referred to as AV with respect to their ancestry. But what we really didn't know yet was whether or not we were looking at the same sex-determining region or if it was something that moved between them. So our whole genome sequencing, however, has provided us unparalleled insight into what's going on there. And I'll just briefly tell you that what we've identified is a 20 kb insertion in the females of Fragaria chiloensis. That's really exciting to us because it has a lot of similarity to the portion of the sex-determining region that we've identified in Fragaria virginiana. And since I'm running out of time, I won't talk too much more about that, other than to show you that they actually share this core feature of that sex-determining region. So we're now able to conclusively say that in Fragaria virginiana, we do have 2 linked loci. In chiloensis, the sister species, we have the same dominance relationships, but greater recombination suppression. But really interestingly, these different chromosomal locations within homologous group 6 suggest possibly that male sterility was initiated once and through at least one translocation moved to a new location in Fragaria chiloensis. This also provided us a really interesting counterpoint to the idea that chiloensis with the greater sexual dimorphism is actually the younger of the 2 species. And perhaps this translocation event helps stimulate that enhanced progression of the sex chromosome within that species. So I just want to quickly finish up and say that I think that plants might provide some really interesting comparative work for understanding sex chromosome diversity, potentially because of their processes of polyploidization and hybridization, which are common in their evolutionary histories. And I just want to quickly sum up by acknowledging that this work was done by many people in both my labs and the lab of Aaron Liston at OSU University, in particular the work of Jacob Tennyson, who's the bioinformatician on this project.